step of cell signaling, we have reception, to where a ligand binds to a receptor protein, and the receptor then undergoes a conformational shape change. The shape change transmits the signal to the inside of the cell. Then we have step 2, transduction. Then the signal is moved from the cell membrane through to the nucleus. And this happens to be through a phosphorylation of kinase proteins, and phosphorylation just means adding a phosphate. Then we have our response. The response takes place in the nucleus, to where the final molecule of the signal pathway initiates the transcription of target genes, and this leads to a protein being made, which causes the cell response.
Cell communication is the process of one cell sending ligands to another. There are two mechanisms of ligand delivery: cell-to-cell contact and paracrine signaling. Cell-to-cell contact is cells making physical contact and transmitting signals and initiate the cell response. The direct contact between signal and receptor proteins bound on cell membranes. This is traditional, but there's also gap junctions and plasmodesmata. Gap junctions are for animals and plasmodesmata are for plants, but they're pretty similar in terms of the cells fuse together and pores open up between them to allow the signal or the ligands to quickly diffuse through and initiate the cell response. Now, the other type of cell communication is paracrine signaling. This is when the communicating cell releases a signaling molecule, the ligand, that moves to the target receiving cell. Ligands can travel short distances through diffusion or long distances through the bloodstream. When it comes to cell signaling, there are two types of ion channel receptors. There's membrane-bound receptors and intracellular receptors. Membrane-bound receptors are for hydrophilic ligands. Basically, the hydrophilic ligands, they can't go through the cell membrane, so they bind to an embedded receptor in the cell membrane. The ligands don't actually cross the membrane. The signal is amplified by the embedded receptor and moved from the membrane to the nucleus via a transduction pathway. The other type of ion channel receptor is the intracellular receptors. This is when a ligand is lipid soluble, meaning it's able to diffuse right across the membrane, and it binds to a receptor inside the cell. And this receptor allows the ligand to bypass the transduction pathway and travels directly to the nucleus to start the cell process.
Then we move to signal transduction, where the membrane-bound receptors require the use of transduction to move the ligand signal from the membrane to the nucleus. The ligand binds to the receptor, causing it to change shape. This transmits the signal into the cell. The second messenger, also known as salicylic AMP, is activated and phosphorylates the next protein in the chain, which activates it. This occurs over and over until the last protein in the pathway enters the nucleus and initiates a cell response. So in this image, we can see the inactive and active proteins being phosphorylated until it gets to a point where it can cause a response in the nucleus and activate the gene. As we move on to G protein coupled receptors, otherwise known as GPCRs, we have an example here that shows an activated receptor and a ligand being attached to it, and then the ligand binds, and then this causes the G protein to phosphorylate, where, as we can see here, the GDP turning into GTP, which basically just means it's adding a phosphate to then go activate the enzyme. So then once it goes to GTP, it travels over to the inactive enzyme and activates it. And this then eventually leads on to the cell response. Receptor-tyrosine kinases are essential components in signal transduction pathways that help with cell-to-cell communication. They're transmembrane receptors which bind to the ligands, mainly growth factor ligands, and play a key part in cell growth, differentiation, metabolism, and motility. So these help absorb the signal by the ligand and then send it down to the kinases to get phosphorylated, which then can cause a response.
Feedback loops and homeostasis. Feedback loops are sensory pathways and responses that the body uses to maintain homeostasis. There are two types of these feedback loops: negative and positive feedback loops. Negative feedback loops slow down or stop response. They regulate physiological processes by stopping the cellular response, and that when that cellular response stops, the body returns to its normal state, which is homeostasis. For example, when your body temperature increases, you start to sweat, and that cools you down, decreasing your body temperature back to homeostasis. The second type of feedback loop is positive feedback. Positive feedback speeds up or amplifies responses, and basically it's responsible for initiating and amplifying biological processes in the body system. It produces large systemic changes to reestablish homeostasis. The main example used for positive feedback loops would be like childbirth, as it's shown in the picture.